Let me clear my throat. Much better. Transcription and protein translation. So this together makes up what's called protein synthesis, right? We're synthesizing protein. We're making proteins. So transcription is just a lot like DNA replication. So what you do is you have your DNA strand, and what happens is it unwinds to form a single strand of RNA. In this case, it's mRNA. It's messenger RNA. Messenger RNA can leave the nucleus. We know DNA cannot leave the nucleus. Messenger RNA can leave the nucleus. So it bounces. It gets out of there faster than I left Grays Lake, Illinois after graduating from high school. I'm out of here. So RNA does the same thing. It leaves. So what it does is it goes into the cytoplasm, into the ribosomes. And that's where this translation really takes place. That's where once it's transcribed, that's when it really does its work in terms of forming proteins. Now we're going to see a word here that we use a lot. It's called codons. It's just a 3-letter sequence. So like AAU, CAG. So anything that's 3 letters would be a sequence, would be a codon. The one thing I do want you to remember about RNA is that other than it, remember RNA is single-stranded. So it's smaller than DNA. And it also has uracil instead of thymine. So why is that? Great question. It's real complicated. Basically what it comes down to is it's just, think of it, it's energy efficient. When you get to the translation part, you're making proteins, our body needs a lot of proteins. So uracil just makes that process easier. If you want to geek out and figure out why, then you can figure that out on your own time. Or come see me and we'll talk for hours and hours on the difference between thymine and uracil. All right, step 1, transcription step 1. Here's our DNA. How do you know it's DNA? We know it's DNA because of the thymine, okay? We know it's DNA, it's double-stranded. You have your phosphate sugar backbone, right? You have your beautiful base pairings. Now this sequence is what makes you. The sequencing of the base pairs is what makes us unique. So because proteins are based on the sequence, our proteins are going to be unique to us. So like replication, you're going to have the, DNA unwind, and then you're going to have an mRNA, messenger RNA. Now there's all different types of RNA. I'm just going to focus on messenger RNA and then tRNA today. So again, you see it's matched up. Hey, look, it's RNA because of the U. Okay? DNA is the T, RNA is the U. Great. So then this thing leaves. It pieces out. Anyway, okay, step 2. So step 2, after that's transcribed, your mRNA, again, leaves the nucleus. Sorry, step 3. mRNA leaves the nucleus and goes to the ribosomes, the ribosomes, if you will, in the cytoplasm, and they have a ribosome, an mRNA ribosome party. It's like a rave for protein synthesis. Okay, so once RNA is transcribed, it leaves the nucleus, transcription is done. We're now moving on to translation, right? Lost in protein translation. Okay, so that genetic code, what we mean by modified genetic code, we mean is that RNA is then translated into proteins. Here we go. We know that proteins are made up of amino acids, right? So the job here is that translation takes amino acids, because amino acids are made up of 3 base pairs, right? They're made up of 1 codon. So a 3 base pairs sequence, like basically codes for an amino acid. So based on that DNA sequence, which turns into that mRNA sequence, that determines the order of amino acids linked together, and we all know amino acids make up proteins. Hope that made sense. So here's another little fun fact in terms of like where the diversity of life comes from is that there are 20 amino acids, each are made up of 3 base pairs, but there's 64 combinations of these codons. So even though there might be 20 amino acids, the different combinations, you're going to have multiple codons coding for the same amino acid, if that makes sense. So you might have like leucine or whatever, and you might have, so one amino acid, guys, I'm going to make this part up. You might have, this might code for leucine, this might code for leucine, okay? So that's just kind of a fact. All right, so you have the mRNA in the ribosome, and then you've got tRNA doing its work. Now, while mRNA, the codon is on mRNA, the anticodon is on tRNA, okay? tRNA stands for transfer RNA. All it does, again, it goes from 5 to 3. All it does, it just kind of like has a 3 sequence here, basically. It grabs the proteins from here and attaches them to the mRNA, which allows it to attach to other amino acids to make a protein. So again, this is what's called an anticodon. It's a 3 base sequence again. It is a protein molecule, and its main job here is to attach the amino acids into the ribosomes to make that protein. So the way protein synthesis starts is that the ribosome binds to the mRNA, okay, at the start. AUG, that's how it starts. Once that happens, so, oh God, I'm going to try to draw this. Here's our ribosome. Here's the ribosome. Here's our mRNA strand, okay? So here's a little strand. So here's our strand right there. And then this is the ribosome. That's step 1, okay? That's the start. Now what happens is the tRNAs, they're shaped like a T. They're called tRNA because they're shaped like T's. They will grab, they kind of are floating out here. They'll grab the amino acids. They're just hanging out, looking for friends. These guys will grab them. Hey, come on, friends, come to this party. Bring them down here, okay? So as they do that, the amino acids link together and form a protein. And it moves, the ribosome moves along the mRNA, okay? And they're added one by one. Finally, something comes along, the cops come and stop the party, and the ribosome and the mRNA separate and go hide behind the bushes until the cops leave. So I've been told that's what happens when cops come. All right, so that's a good time. So here's an overall big picture. Hopefully this makes sense, guys. If not, please bring in like 2 to 3 questions so we can go over them. I know the translation part is very confusing because you've got your mRNA here, you've got your ribosome here, you've got the tRNA, you've got codon, anticodon. But I think the nice thing about protein synthesis is that, or at least the translation, is that it's much more visual. We kind of have a visual idea. So hopefully this kind of makes more sense. If not, bring in your questions and I will, guys, I'm going to answer them. I know it's my week. I know it's Teacher Appreciation Week, but I mean, I just can't. The true joy, the true gift is, being able to construct knowledge with you every day. I was not struck by lightning somehow after I made that statement. All right, bring in questions. We'll talk to you tomorrow. Great. Peace out.